Welcome to the DECODON webinar, part 2 of the webinar from image fusion to publication. Today we will talk about how to export Picklists from 2D GIS, how to re-import MASBEC data, to run web queries, and how to report results. The core topics that we will cover today are how to create labels and how to manage them, to move and copy them to other images, how to export pick lists for interesting spots, how to replace existing labels with maybe spot IDs by the MASBEC results, namely the protein identifications, how to run web queries based on protein identifications to have access to more detailed data coming from web resources, and finally how to report the results that we have from our MASBEC workflow. Delta 2D includes a recommended workflow which has 5 steps: the setup project step, the warp images step, then detect and quantify spots, followed by analyze expression profiles, and then finally the step to present results. Today we will move in the step present results. This part of the workflow in a more detailed view. Imagine we have found spots of interest already. We can automatically label them with either numbers or with spot IDs. We can create pick lists for the labeled spots, for the interesting spots, and then outside of Delta 2D, of course, you can take the pick lists, pick the spots of interest, and then come back with spot identifications and use the translate label feature in Delta 2D to replace the automatically created labels, including numbers or IDs, anonymous labels, by the protein identifications. Based on this, you can employ scouts to query web databases of your choice to add more detailed information to your protein spots and then visualize the results and report your findings. The starting point of these steps is that we have already complete expression profiles and have reduced the number of expression profiles to those of interest. Having complete expression profiles, as you might remember from previous webinars, are very important to reduce the problem of empty areas in the quantitation table, of missing values. Missing values decrease a lot the statistical significance of your findings, and that means having no missing values increases the statistical significance to a maximum. Having complete expression profiles is not enough. We also want to focus on interesting ones, and this image here is just a symbol for doing statistical analysis to have only interesting expression profiles then. First, in this webinar, we'd like to create labels for the spots of interest. So you can label each spot separately, every single spot on a certain image, but you can also label all the selected spots, all the marked spots, or all the not yet labeled spots at once. Doing this automatically means you just attach spot IDs or spot numbers as labels to the spots. This can be also completed by using a prefix. You can define a certain string which is then followed by the number. You can also move or copy labels between images. That means you can just take a label, and if you want to pick this spot from another GIS, just move this label to the other image, and then you have this available there, of course, with respect to the image warping, so that it's really at the proper position. Let me show this in Delta 2D. Here we go. This is the demo project in Delta 2D, the simple demo project. It contains 3 groups: the control group with 2 replicates of a control sample, the 1-minute group containing 2 replicates of a sample taken after 1 minute, maybe cell growth, and 1 group called 10 minutes, including 2 replicates of the sample taken after 10 minutes. Then we also have a group including the fused image, which has been created by when working through the recommended workflow in Delta 2D. Let's have a look at the control 1 image. Here we go. This is the image. The contrast settings are automatically optimized for this image so that we really see all the spots. And we can now activate the label tool here and click on a certain spot so that this spot is then annotated. For example, with this prefix int.spot underline and then a consecutive number. The next spot is then in spot 2. Of course, you can click on even more spots to just manually assign numbers to them, and we can change this prefix, for example using the options, going to the labels tab and then changing the prefix from int.spot underscore to spot underscore, for example. With this you can create different subsets of labeled spots. For example, you could use up-regulated as a prefix or down-regulated as a prefix so that you number the up and down-regulated spots separately. So if I now click again on a spot, it is spot 3 with the prefix spot underline again. I can right-click on a certain label to delete it or to move or copy it to the other image which is involved in this dual view here. We can also assign labels to a set of spots. So let us open a table for this image. Let me select this image again and open a table only for this image now. We have already prepared a set of spots to be of interest. We have marked them, and let me now select all these spots and go back to the dual view. Let me show that now all these selected spots are also highlighted in the dual view. The views are synchronized, and now we can just define labels for these selected spots. I choose the labels menu, and then I could label these selected spots with spot IDs or just with numbers. Let me assign numbers to the selected spots on this image control 1. As you can see now, all the spots have their label now. You can move label texts to wherever you want on your image, and you can also move the complete labels if a label should show to a different spot if you misclicked on a certain spot. Having labeled the spots, we can then export pick lists for these spots of interest. Delta 2D supports a variety of spot pickers, and it could happen that in the time that you have taken to analyze your images, the gel has been changed. It might have been dried, shrinkled, whatever, so that the spots have moved slightly. You can avoid this by using glass plates or transparent foil-based or backed gels, but usually your gels will change, and that means that the spot positions on your analyzed image do not match the real positions of spots on the picker. For this reason, usually a picker has an imaging device, and then you can just take a new image from your gel as it is positioned on your picker, import this new image into Delta 2D, warp it to the original, the analyzer's image of this gel, and then copy all the labels from the analyzer's gel to this picker image. And that means that then the labels are attached to the real positions of spots on your gel as it is appearing on the picker. If you don't have a picker, you can also use a generic Picklist format just to export the list for manual picking, for example. The Picklist includes typically an ID, the X and Y coordinates, and usually also the label names that we have assigned to the spots. And you can export picklists that include the labeled spots, the marked spots, and also the labeled positions, which means that you do not have to detect spots on your images at all. You can also just qualitatively analyze your images and then say, I want to pick this spot and that one, and you would then pick them after having just labeled their positions without having a spot detection or even a quantitative analysis done for your images. Let me show a sample Picklist. It looks like this. This is just a text file, flat text file, including character separated values. Character separated means that the different columns are separated by, for example, a tab or a semicolon or a colon, whatever. And in this case, we have in the first column the spot ID, in the second one the X coordinate, in the third one the Y coordinate, and then this is followed by a fourth column including the spot labels. Let me show this in Delta 2D. We have prepared another image where we have real interesting labels, in a total of 29 labels, and then I choose the export menu, I choose export picklists, and then I could now choose the picker that I have in my lab or the generic format for this image control 2. A dialog opens giving me access to my folders, to my file system, and then I can save the picklist to a certain position or a network drive where it should then be picked by the picking control software, where it should be imported, and the picker then picks these positions and puts the different spots into different wells. And then you can continue with the workflow outside Delta 2D, do the mass spec, or let someone do the mass spec, take the mass spectra and then searching for hits, and then you come back with protein identifications. Having these protein identifications, the next step is then to replace the original protein numbers, the labels, the anonymous labels just containing numbers of protein or spot IDs, by the real protein identifications. That means what we need is a small table containing the previously defined label and the appropriate respective protein identification. And then we can import this table, and then, for example, spot 04 would be replaced by ANO, and ANO would then be shown on the jail image instead of our anonymous label then. Such a translate table should look like this. In the first column we have the old label name followed by a character that separates the values again. It could be a tab, as in this example, or a comma or a semicolon, and then this is followed by the protein identification for this spot. Let me show this in Delta 2D. We have these labels on our image, and now we want to replace them by the protein identifications. I choose the labels menu, choose translate label names for my image control 2, and then I get a dialog where I can choose the character that is delimiting the different label names. I choose the tabulator, and then I can load my translate list, and then I have a preview of how my old original label names would be translated into protein identifications. You can scroll down and see that, for example, spot 4 in this example is replaced by GrowL. Now it could happen that your identifications do not include all the spots, that sometimes spot identification has not been possible. And then we can decide whether we want to remove the labels that do not have a translation or if you want to keep them. Pressing OK means my old labels are now automatically replaced by these new protein identifications. Based on these protein identifications that I can see on my image now, you would typically then create a list of your identifications and maintain a certain table. You would take for each single row the protein identification, do a query, for example in UniProt, submit the query and get back different attributes, for example functional categories, the protein sequence, the isoelectric point, molecular weight, the number of amino acids, whatever. And then you would have to copy and paste all these attributes from the response of your web database to the table that you maintain. And this is not only very time consuming, it's also tedious, it's no fun, and it's also erroneous. That's why we thought it's good to create automatically the queries by DECODON scouts. So for example for UniProt you would just submit a query automatically for the label, and then UniProt is answering to Delta2D and provides the attributes automatically, which will be then attached to your label. In the second step, Delta2D is taking then the protein sequence, is sending it to Emboss and gets then back the isoelectric point and molecular weight for this specific protein sequence. This is then looking like this: isoelectric point, molecular weight for this theoretical protein sequence as it came from UniProt. If your favorite database is not included in the list of supported scouts that we have up to now, for example, it could be that you want to use a certain very organism-specific web database, then you can just develop your own scout or let us do it for you, since the DECODON scouts are based on an open architecture and additional scouts can be added rather easy. It's even more easy if your web database, if the database of your preference is supporting XML replies. Let me show this in Delta2D. We have these labels now. Let me, with the activated label tool of course, right click on one label and then choose Edit Scout Data from the context menu and then the UniProt Scout. The protein identification is then used as a default input for the query and the organism is taken from my last query that I have submitted. I press then Get Data, and now Delta2D is submitting this query and UniProt is delivering the attributes that we want to see. For example, the gene name, the sequence of the protein, the amino acids, and so on. And here we have the isoelectric point, which is not taken from UniProt but from Emboss. Gene ontology and all these blue strings are links so that you can easily open the website for this protein in UniProt, for example. We do not have to run the query for every single label. We can do this for all the labels in a batch. Let's choose the labels menu again and then search for Fetch Scout Data, choose the UniProt Scout for our image control tool. Then I have the choice whether I want to overwrite the existing results from previous scout queries. I say no, and then I can also choose a different organism name for this query, and I say OK, and then Delta2D is submitting for every single protein identification a query to UniProt and is collecting the answer for this query. This takes a second. And again, let me stress that with these queries are automated and you won't miss a single letter, for example, of protein sequences, which can easily happen if you copy and paste this manually from your web browser to the spreadsheet calculation software. So we not only want to save your time, we also want to reduce the sources of possible errors. Of course, the queries take a second. We, of course, depend upon the speed of UniProt here and the internet of connection, of course. So this is now done for all the different labels that we have. Let's have a look in one example. I right-click again with activated label tool, of course, on a label, can edit scout data for UniProt, and then I can review the data that has been coming from UniProt for this label. Having all these information, for example, from UniProt, we can use quantitative information to, for example, color the protein identifications, the label texts on my gel image. For example, here the PI, the isoelectric point of a certain protein sequence, is taken to define a color gradient across the data range of this PI, and then coloring beginning left with blue, ending with red on the right side, the different PIs with different colors. And if we zoom in, then we can see that some labels have a different color compared to their neighborhood. We would expect orange here, but get a certain shade of violet, and that means the theoretical isoelectric point calculated for the protein sequence that we received from UniProt does not match the real position on the gel. So in this case, the protein sequence that we have received from UniProt is not the same as it appears in the gel. Maybe there's a modification, an isoform, whatever, and then you can follow up this, run another search with maybe a more narrow PI range that you're looking for. Let me show this in Delta 2D now. We already have the labels, the protein identification, we already have scout data, attributes that we have received from UniProt. Now let's just color these labels by the color gradient for the PI. I choose labels, choose the formats entry of this menu, and then instead of defining this color for the text automatically, I choose a scout for input, as an input for the coloring. This dialog has detected that there is only one scout including quantitative data, and then I can choose the property that I want to use to color the labels. I use the isoelectric point and define the rainbow color gradient to have a very fine-grained change of colors across my gradient of PI. I choose OK and OK again, and then you can see that the text is automatically colored, and we also see that there is also in this example an outlier. For example, this spot GLNA has a green color, while all the others here have orange or maybe a more yellow or red color. That means you would now follow up this protein spot to have a look what happened with this protein. Having done this, we can then report our findings, for example, as a spot album to show a certain area on the images, the area of a spot of an expression profile across all the images, or for example the label scout including the label report including scout data, for example from UniProt, like this. The reports are very flexible. You can configure the output. The reports are still connected to Delta 2D so that you can change what you want to see in a report. You can open multiple instances of reports at the same time in different tabs of your web browser. They are HTML based so that you can also easily define the design changing the custom style sheet and replacing, for example, our logo by your institute's or your company's logo. And of course, you can easily copy and paste the content of the report to any other application. And finally, you can also, since these reports are web-based, every single report has a unique URL. You can copy this URL and insert it to your spreadsheet calculation software like Excel, OpenOffice, or LibreOffice to have access to the tables that are included in the report automatically and import the tables into your tables in the other application. Let me show this in Delta 2D again. In the main menu we have a menu item called Reports. It includes an index, an overview of all the reports, but also access to the 4 different reports. I open the index and then we see there's a project summary, a spot album, a spot quantity, and a label report available. Let us first have a look into the project summary report. This report includes just descriptive information about, for example, our groups, the images in the different groups, some attributes of the images, how many spots are available on the images, and so on. And also information about the samples, if you maintained this information in Delta 2D, and information about the warping. And we can also click to report, and then all the report as well as all images with an applied contrast setting as defined in Delta 2D. And this report can now also be used not only for reporting but also for quality control. For example, after the image warping step, which you might have delegated to another colleague, you tell them, before going to the next step and fusing the images, let's have a look into this report, and then you say, okay, in a certain region, maybe improve the warping here or there, and afterwards let's continue with the image fusion. Let's go back to our index of the reports. There's also the spot album report. This comes with a small form. We can choose whether we want to see selected or marked spots on a certain image, for example on the control 2, and then we can also, among other settings, control the size of the neighborhood of our spots that we want to see, and also a zoom scale for these tiles that we have. I apply these changes and get this report for our marked spots on control 2. As you can see, there is the region around this spot across all the images, and you can then see it's headed by the bar chart, and then you can just see whether the quantities are also visible in the gel images, that the intensity of the spots really is measured by the bar chart in a proper way, like this. And this is then the list of interesting expression profiles. If I click on one of them, we see that there is a detailed report for this expression profile. Again, the bar chart and also the images, but also quantitative information and a nice view on a certain gel image where we can see this spot highlighted so that we see on which position this spot is appearing on the gel. Let's go back to the index and show the next report, spot quantity report. Again, we can choose whether we want to see the selected or marked spots on a certain image. I change this report to apply my settings, and then we have a nice condensed view with just statistical data for my interesting expression profiles, but I can also choose to have a look on a wide view, and this then contains a large number of columns. All the columns available in the different tables in Delta 2D are now shown here, and then you can just take this table and import it to another application, delete the columns which are not of interest then, and continue your analysis. We also have an image report which means where we see all our, in this case, marked spots marked on a certain image, and then see them on the image itself, so that we can have all them highlighted on an image. We can click here to see the detailed report for this one, and then also see it here again, the spot. Let's go back to our report index and come to the labels report. This report shows us the labels on a certain image, for example on control 1. Here we go. And if I choose, for example, those on control 2, then also see that these labels not only have a certain identification in name, but also UProt data, since we have done these web queries for them. And this report now includes, for example, also the protein sequence and the UniProt entry again as a link, so that you can easily now go to UniProt and have a look on additional attributes that are available in UniProt for this protein. Let me also mention that there are two columns here, the estimated PI and molecular weight. They are estimated based on the position of this protein, and input for the estimation are the available theoretical values as they came from MBOSS. So all these isoelectric points and molecular weights are then taken as an input for a calibration and estimation of the real PI and molecular weight. And then you can see, okay, the PI of 4.11 is slightly different to 4.2224, but maybe not too much, while maybe another one is too far away from the theoretical molecular weight or PI. So now we are through the aspects that we want to cover in this webinar. Thanks for your attention. Also, thank you for using Delta2D and for the different publications that we had already. If you have a publication which is not yet included on the reference list on our website, let us know. Send us your publication and we will add it there. And also, we have seen a lot of different proteomics covers which have been very nicely designed with images taken from Delta2D. Let me also remind you that DECODON has had its 12th anniversary recently. For this reason, we offer a 12% discount for all the orders that we receive until 12th of December this year. Use this possibility to order your update, maintenance for another year, consumable credits, or the refraction 2D dyes, which enable you to run DIGE experiments. Let me also invite you for the upcoming webinars next year's. You can have a look on our website in the section Academy and then Webinars. We will show upcoming dates. For the complete year 2013, we plan to publish the dates in January. If you have any other questions, let us know. Send an email to support@decodon.com or give your sales rep at Decodon a phone call or send them an email. Again, thanks for your understanding. We are looking forward to meeting you in one of our next webinars. Bye bye.